hormone we move on into is cholecystokinin. Cholecystokinin is secreted by eye cells in the duodenum and jejunum. The stimulus for its release is basically fat and protein entering the duodenum. So whenever you have a fatty meal and it goes into the duodenum or basically the small intestine, the main stimuli that leads to cholecystokinin being released is the fatty acids or the amino acids and peptides from that fatty meal. Now, the main function of cholecystokinin is basically to contract the gallbladder. Contracting the gallbladder releases bile into the duodenum. Why? Because this helps digest fats. And the other thing that cholecystokinin does is relaxation of the sphincter of Oddi. Now, obviously, sphincter of Oddi is that sphincter that stops the pancreatic duct and the bile duct from entering into the duodenum. Relaxation of the sphincter allows bile and pancreatic enzymes to flow into the intestine. Another thing that it does is the pancreatic enzyme secretion. The cholecystokinin stimulates pancreatic acinar cells to release digestive enzymes. Examples of these enzymes are trypsin, chymotrypsin, lipase, amylase. These digest proteins, fats, and carbohydrates. Cholecystokinin also slows gastric emptying. Why? And it also delays stomach emptying because fat digestion needs time. Since fat digestion takes time, cholecystokinin delays the stomach emptying so it can be digested. It also gives a satiety signal. So it gives a satiety signal to your brain that I'm full. Cholecystokinin acts on the hypothalamus and the vagus nerve, and it causes a feeling of fullness. This is why fatty meals make you feel satisfied longer. So cholecystokinin is basically the fat digestion hormone. It helps to contract the gallbladder, and pancreatic enzyme secretion also is done through it. Basically, it relaxes the sphincter of Oddi and it slows gastric emptying and it makes you feel full. Now, whenever you get gallstones, this bile stasis. The bile slows down and stays there. Now, whenever cholecystokinin signaling is impaired, the gallbladder doesn't contract well, and that leads to stasis of the bile. So it slows down and stays there, and that leads to gallstones. So whenever cholecystokinin signaling is impaired, you get gallbladder that doesn't work well, it doesn't contract well, and the bile is stasis, and you get gallstones. Another thing is the cholecystokinin stimulation test. This is used to diagnose biliary dyskinesia. The cholecystokinin stimulation test checks how well the gallbladder contracts when stimulated. So basically, when you eat a fatty meal and the fat enters the duodenum and the eye cells release the CCK, the CCK causes gallbladder contraction and relaxation of sphincter of Oddi, and the bile flows into the duodenum. So if the gallbladder does not contract properly, bile does not empty well. This condition is called biliary dyskinesia. This is a functional disorder of gallbladder motility where the gallbladder fails to contract normally even though no gallstones are present. So typical symptoms would be right upper quadrant pain, pain after fatty meals, nausea, bloating. Ultrasound shows no stones. We suspect motility problem. So you basically give a radioactive tracer injection, um, and and then the gallbladder fills, and the gamma camera tracks this. And then the cystic duct is open and gallbladder is present and functional. So the synthetic CCK is injected, and this mimics a fatty meal and should cause the gallbladder contraction. And then you measure the gallbladder ejection fraction, right? And then you see if this is normal or not. So that's something that is used for clinical correlation of cholecystokinin. Other than that, there's also something to know is that if pancreatic enzymes or bile do not reach the intestine, you get steatorrhea, and steatorrhea is basically fat malabsorption. We now move on to secretin. Secretin is the hormone secreted by S cells of the duodenum. It is released when acidic chyme enters the duodenum. So now whenever you have the acidic chyme entering the duodenum, you get the secretion of secretin from the S cells. So what this basically does is the action of secretin is basically an increase in pancreatic hydrogen carbonate secretion. So it stimulates bicarbonate secretion. The pancreatic duct cells make the bicarbonate, and the purpose of this is to neutralize the gastric acid entering the duodenum. Then another thing is gastric acid secretion is decreased. This is again to protect the duodenal mucosa, right? The liver basically releases an alkaline bile. So the bile secretion is increased, and this alkaline bile also helps to neutralize the acid pH. Secretin acts as a brake on the stomach. So the gastric acid secretion is decreased because secretin acts as a brake on the stomach, and this effect leads to gastric acid being decreased and gastric emptying being decreased. Okay, so summary is that the secretin neutralizes acid. It is regulated by an increase of acid and fatty acids in lumen of duodenum, and the hydrogen carbonate neutralizes gastric acid in the duodenum, and this allows the pancreatic enzymes to function. Otherwise, the pancreatic enzymes would not function in an extremely acidic environment. We now move on to the glucose-dependent insulinotropic peptide. This is one of the incretin hormones that link the gut to the pancreas after a meal. It's the source that it's secreted from is basically K cells in the duodenum and jejunum. So these K cells release this GIP, the glucose-dependent insulinotropic peptide. Whenever food enters the small intestine, that's when these K cells release the glucose-dependent insulinotropic peptide. The stimulus for release is glucose, fatty acids, and amino acids. So you can see that it's increased by fatty acids, amino acids, and oral glucose. So whenever nutrients enter the small intestines, the GIP levels rise. The main function is basically there are two functions. So there's an exocrine function where there's a decrease in gastric hydrogen secretion, and then there's an endocrine function where there's an increase in insulin release. So basically, insulin secretion depends on the glucose level, right? And this is why it's called glucose dependent insulinotropic peptide. So if glucose is low, GIP does not stimulate insulin much. But if the glucose is high, the GIP stimulates the insulin secretion. And why is it called an incretin? Basically, incretins are basically intestinal hormones that increase insulin secretion after oral glucose. We have two incretin hormones. We have the GIP, this glucose-dependent insulinotropic peptide, and the other one is the GLP. Okay, GLP-1 is glucagon-like peptide 1 agonists, and these are a class of medications that mimic the natural incretin hormone to manage type 2 diabetes and obesity. Okay, so we use these GLP receptor agonists like medicines also. But other than that, a GLP is basically a glucagon-like peptide one. So this hormone can be used to manage diabetes and other medication, other like obesity diseases. Otherwise, this GLP-1 is a natural hormone. It's produced in the gut and it regulates blood sugar and appetite. It's basically produced by the intestine, mainly the ileum and colon. They produce it after meals to stimulate insulin, inhibit glucagon, which basically raises blood sugar, and signals satiety to the brain. And that's why the GLP-1 medications, which are basically agonists, strengthen these signals more, and that leads to improved blood sugar control and significant weight loss. Also, it's also this glucose-dependent insulinotropic peptide is also known as gastric inhibitory peptide. Why? Because it can decrease gastric acid secretion and it can slow gastric motility. So it's also known as gastric inhibitory peptide. But its major physiological role is in insulin secretion whenever it counters an increase in amino acids, fatty acids, and oral glucose. And oral glucose load is increased with insulin compared to IV equivalent due to GIP secretion. So if I give a patient or a human an IV load of glucose and I compare the insulin secretion with the oral glucose load, I will have an increased insulation in the oral glucose load compared to the IV glucose load because of glucose dependent insulinotropic peptide. And that's why it also has a role in diabetes. In type 2 diabetes, the GIP is produced normally, but beta cells don't respond well to it. So the incretin effect is reduced. This is why drugs that enhance the incretin activity are used here. And that's when we use GLP-1 receptor agonists, which are basically semaglutide, liraglutide, exenatide, and all of these increase insulin. They decrease glucagon, they decrease gastric emptying, and they decrease appetite. Okay, we also have some DPP-4 inhibitors. The DPP-4 enzyme normally breaks down incretins, right? So drugs that block the DPP-4 enzyme are like sitagliptin, linagliptin, saxagliptin, and this results in increased in glucose-dependent insulinotropic peptide, and the other one, GLP-1 is also increased by this. So basically, it leads to increased insulin secretion, and that's why DPP-4 inhibitors and GLP-1 receptor agonists have a very major role in diabetes treatment. We then move on to the hormone Motilin. Motilin is a GI peptide hormone. This controls the intestinal movement during fasting. It's especially important for the migrating motor complex that we will study in a bit. The source of Motilin is basically small intestine. Okay, so this produces migrating motor complexes, and migrating motor complexes basically are stimulated by the motilin. Okay, so this is a wave of strong peristaltic contractions that basically move from the stomach and then the duodenum and then the jejunum and then the ileum. So this migrating motor complex is basically these strong peristaltic contractions that move from the stomach, duodenum, jejunum, ileum, and these contractions sweep the gut clean. So its job basically is to do gut housekeeping when you're not eating. Motilin activates strong contractions that clear leftover food, mucus, and bacteria from the intestine. It's released during fasting about every 90 to 120 minutes between the meals. And migrating motor complexes basically occur between the meals. Their purpose is to clear any food debris and prevent bacterial overgrowth and maintain intestinal motility. So you can see that it's increased in fasting state between meals. You have motilin receptor agonist, example erythromycin, which is an antibiotic that is used in constipation. Why? Because it's used to stimulate intestinal peristalsis. So erythromycin is an antibiotic which is used as a motilin receptor agonist. It means it can stimulate motilin receptors and increase GI motility. It is used in gastroparesis and delayed gastric emptying. Example would be diabetic gastroparesis. So what happens if MMC or these migrating motor complexes are impaired? Possible effects could include small intestinal bacterial overgrowth. We call it SIBO, small intestine bacterial overgrowth, or we can have bloating and malabsorption because bacteria are not cleared properly.